We can't go into every experiment that leads to the knowledge that we have about biology in this class, but some are just worth it. And the Hershey and Chase experiments are really great. It was a clever experiment, great technique, and it led to a really important discovery. So I want to explain this one briefly and walk through it. It actually reviews a lot of great biology and introduces us to viruses, which we've kind of left them understudied in this class. Clearly they're important to human health. But let me set the stage. So their experiments were done in 1951, 1952. The next year, in 53, Watson and Crick announced the description of the molecule DNA. And we know how important that was in the field of biology. But people still weren't sure. Was it proteins or DNA that people and other organisms inherited to give them instructions on how to basically make and operate a body? Most people were still in the protein camp. Now, there had been some experiments before that were really clever and indicated it's probably DNA, but this one really solidified it. And they looked at some pretty basic chemistry. They knew that viruses were made of a protein coat and DNA, or sometimes RNA, inside. And they knew that they attacked other cells, and in this case they looked at bacteria, and they hijacked that cell. Basically they took it over, rewrote the instructions and said, hey, make more viruses. That's what they do. So they completely took over the operation of a cell. And they figured whatever this injected into that cell must be the molecule of inheritance. It was giving it new instructions. And they only had two options, so this was a perfect setup. Here's the chemistry. So in terms of the chemical composition of proteins, you have carbon, hydrogen, oxygen, and nitrogen, and very often, most of the time, sulfur as well. Not a ton, but a few amino acids, or a couple of them, have sulfur. DNA, made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Notice that all those atoms, all those elements, are in common with the two except for sulfur and phosphorus. Well, if you wanted to label something, you label the beaker or petri dish or your pen, you just put your name on it. But you can't really do that with an atom. But you can. You can get a radioactive version of that atom. And so they worked with radioactive sulfur and radioactive phosphorus. What that did was it made DNA radioactive, or it made the proteins radioactive, depending on which chemical they used. If they used radioactive sulfur, it made the proteins radioactive. If they made the phosphorus radioactive, it was the DNA. And then they could track results. Here's what it looked like. Well, here are our intrepid researchers, Alfred Hershey and Martha Chase, back in the 1950s. And here are their research organisms. So we had bacteria and we had viruses. And there's really just two rounds of this, like I was saying. This one was radioactively labeled sulfur, so it's sulfur 35, one of the isotopes of sulfur, and that labeled the proteins. And you can see here, it's that red is showing that that's the protein coat. They didn't know that at the time, that it was the coat of the protein, just because they couldn't see with that much detail. These guys were labeled with radioactive phosphorus, that isotope, which labeled the DNA. So they knew they were labeling protein and DNA differently. And so they grew up a bunch of viruses with radioactive sulfur, let them infect the bacteria, and then kind of gave them enough time where the virus and the bacteria separated. And they did the same thing down here. So they grew up this batch of viruses with radioactive phosphorus, making sure it coated all the DNA, gave them enough time to infect the cells, and then allowed them to separate. And let's look at what happened next. All right, so that was the first part, getting those bacteria infected. The next part then was to separate out the viruses and the bacteria. And they did this with basically a tabletop merry-go-round that spins super fast. It's called a centrifuge. And you put these test tubes in it. They're full of bacteria and viruses, and you spit it really fast, and the heavier stuff settles to the bottom. And so all the bacteria settle out at the bottom. And of course, those bacteria would have been carrying whatever it was that the virus injected into them. So, question is, you had a batch grown up with radioactive sulfur. That was the protein component. You had a batch grown up with radioactive phosphorus. That was the DNA. And so to figure out what was injected, was it DNA or was it protein? I gotta know. You take your little probe for radioactivity, the little Geiger counter, the one that goes tick, 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 tick. And what they found with sulfur was that all the radioactivity was up in here. That's where the viruses still were. Viruses are really small, so they don't settle out as much as the bacteria do. But when it came to the radioactive phosphorus carrying the DNA, the radioactivity was all showing up down here where the bacteria were. So it must have been that DNA with the radioactively labeled phosphorus made it into the cells, and that's why they were seeing radioactivity down there. An elegant experiment. Is it protein or DNA? Well, let's label them with some radioactivity and find out. And the next year, Watson and Crick were set up to say, DNA is the molecule of inheritance, and here's what it looked like. Some great science there.